Oh, hi all. Welcome to the live session of week 8 of Animal Physiology lecture course. So, I am Harsha Shankar. I am a fourth year PhD scholar in Department of Biological Science in ISER Berambur. I am working here in Ribonucleoprotein Biology Lab under the guidance of Dr. R. Selve Bharadavitru. I am also one of the TA for the course of Animal Physiology taken by Professor Mainakdar. So welcome to the live session of Week 8. So last week we get to see some more aspects of neural system. Neural system is a long class, like we are going ahead one after the other. So if you remember, we started with in lecture one from basic introduction to the hierarchy of organization, um, different starting with the cell, tissues, organ system. Then we went ahead with different organ systems, starting from integumentary system to skeletal system, how the skeletal system coordinating with neurons. Then we went ahead with neurons, structure of neurons, how neuronal activity, then how action potential generate, how the electrical signal gets transmitted, what are the different types of ion channels. Then to after neurons, we get to know what are the different disorders of neurons. So in the last week, we were mainly discussing about different disorders of neurons like Alzheimer's, Parkinson, spinal cord injury, amyotrophic lateral sclerosis, all these we have get to know. Which part of the brain is getting affecting, where is it occurring, what is the disorder, what kind of neuron is getting damaged, how this is getting and leading to this kind of condition, and some type of therapies involved in some of the disorders also uh, discussed. Now we have reached eighth week where we are discussing with stress reflex arc circuit. So in the first three lectures, I was mainly taught us about the stretch reflex arc circuit. Starting with remember when we studied about skeletal system also, we have went through a brief intro of what is stretch reflex arc circuit. So how the muscles are getting coordinated. In that, we mainly look in the aspect of how what are the different molecules present in the skeletal muscle, actin, myosin, how they are able to do all the activity in coordination, and we have so some aspects of neuron coming in that playing a role there also. So here we are talking mainly now we have seen neurons and both skeletal system. We are again discussing how the total stress reflex circuit is maintained and coordinated. So in the first lecture of week 8, sir taught about us mainly about types of reflexes. There are in some case there is direct sensory neuron which takes the information and give it to the central nervous system, spinal cord or brain. Then through motor neuron. So sensory neuron and motor neuron are directly linked. In that cases, we generally call it as monosynaptic reflexes, and in some reflex cases there is one more neuron in between sensory and motor neuron is involved, which are known as interneurons. So if one or more than one type of interneurons are involved, then it is called as polysynaptic reflexes. So remember always, sensor takes the information, pass it to sensory neuron, then it will give it to motor neuron, effector. That is monosynaptic reflex. And in between sensory neuron and motor neuron, one more interneuron is coming. That means that is polysynaptic reflex. Okay. Then Sir gave also a little bit introduction about uh, stretch reflex arc circuit. So remember when we learned about muscle system, we get to see in the muscle there is a sensor present, muscle spindle or interfusal fibers present, which can detect the change in the length of the muscle. Whenever there is a lengthen change of the muscle, that lengthen change of the sensor can be detected by the interfusal muscle fibers. Then in the lecture 2 was the continuation of the lecture 1, where we more talked about extrafusal fibers and intrafusal fibers. Extrafusal fibers are the most abundant part of the muscle, and like they have increased area of the portion, and intrafusal are, as we discussed, they are the muscle spindle fibers or sensors which can detect the change in length. Now the question is, what are the neurons that is connected to these different part of the muscle fibers? To extrafusal fibers, there is a class of alpha neurons coming and getting connected. And to intrafusal fibers, there is a class of gamma neurons coming and taking the information, connecting there. So total information, whatever reaching will be sum of S alpha plus S gamma. Okay. So then we sir also gave us a like a bit of discussion about what is mechanical stimulus and electrical stimulus. So mechanical stimulus is whatever the action that is happening in the muscle after detected by the intrafusal fibers, and electrical stimulus is the all coordinated activity of neurons plus muscle. So MEMS means micro electromechanical system. Then sir started talking about spinal cord injury. We have discussed in the last week. Then sir went ahead with lecture 3, which was the final lecture regarding the structure reflex arc circuit, uh, where he actually talks about what happens in at the end of the neuron. So neurons, multiple neurons are coming and connecting to the skeletal muscle. At that part, we know neurons have some neurotransmitters, especially the muscle which connects to skeletal muscle. We can see the majority of neurotransmitters will be acetylcholine. At the end of the axon, where the before the axon ends, we can a lot of synaptic vesicle which stores the neurotransmitters, and whenever the information goes coming and neurotransmitter will get released as a result, postsynaptic part, that is our muscles binds with the muscle has some certain receptors that can bind and detect these neurotransmitters, which is acetylcholine. And sir also talked about two important such receptors, which are calcium sensors, RYR, rhodine receptors, and dihydropyridine receptors. These are very calcium sensors. So when they open and see, they are directly connected to sarcoplasmic reticulum. As a result, calcium will start opening, and all the different action that we have that we have studied in the skeletal muscle part will now happens. So that time we didn't know that how this calcium is opening, who is telling when calcium to open and how calcium opens, then how actin myosin contraction, everything, sliding filament theory occurs. Now we know how calcium is getting open whenever the delta change in length of the muscle is detected, then that information is connected to the neurons. That neurons will come, acetylcholine will open, it will bind to the receptors RYR DHPR, which will open the calcium channels. Yeah, so that was about the brief summary about first 3 lectures. Let me present the screen. Yeah, so this was we discussed already about first 3 lectures. Stretch 1, we have seen what is polysynaptic reflex, which is mediated by interneuron, and this is a monosynaptic reflex where is mediated without interneuron. It's direct connection with the sensory neuron and motor neuron interaction. Then we started with stretch reflex arc in the second class, where we majorly again talked about extrafusal fibers, intrafusal fibers, their function, and what kinds of neuron innervating what. So remember this part, okay? So always remember intrafusal fibers innervated by gamma motor neuron, alpha motor neuron innervate with extrafusal muscle fibers. Then in the lecture 3, mainly what are synaptic vesicle, and there is nerve action potential after reaching acetylcholine release and binding of acetylcholine receptor with the receptors such as RHR and DHP. And as a result, the calcium ions stored in sarcoplasmic reticulum will get opened, and all the sliding filament theory will can occur. That is was the finish of stretch reflex arc circuit. In the lecture 4, we are now starting with the special senses. So there are different types of, mainly different types of senses, right? Hearing, vision, through the electromagnetic of light wave. In hearing, we can detect the sound or pressure waves coming. Taste receptors can detect the taste. The olfaction can be detected by nose. So all these are different types of senses. So how the ear is able to detect the sound wave or pressure wave, how ear is coordinating with neural system, and how we are getting to know, okay, there is some very heavy sound or there is some sound, someone is talking to me, something is getting detected through the system that we get to see what is the structure of ear. There are three parts: external ear, internal ear, middle ear, and internal ear. External ear is consisted of two mainly part. The outer ear that we are seeing here is pinna or external ear, which consists of, which is continues to a canal, that is the ear canal. Then in the middle ear we can see three soft bones called as malleus, ingus, stapes, which again gives to connect with inner ear, where is the cochlea, all the hair cells. So Shar drew the structure of hair cell, how there is a um, one long hair cells and the adjacent small ones are present. Kinocilium is the longer one. Smaller ones are periothelium, where are labeled it like wrongly in that picture, but we will discuss it in the questions. Okay. So these hair cells are there. So whenever some vibration occur, this hair cells will starts hitting, and as a result, the vibration or waves get hair cells will get coordinated movement. As a result, in the cochlea, that information can pass to the auditory nerves and thereby to the auditory center of the brain. And other than the cochlea, there is one more part inside that, which is the area inside the ear canal, which coordinating with the balancing of the our body. Okay, so ear function is that. And finally, in the lecture 5, sir was talking about the olfaction system. So what is olfaction? The power of detecting smell, right? So whenever there is a strong smell or bad smell or any type of smell, we are able to detect it. How? Because that smell, there is also some kind of receptors and this thing which can detect that. And sir also talked about the strong sensors that is present in the antenna of the insects, how they are this much powerful. And odorants and repellents, the use, the application in agriculture industry for using it against insects of odorants or repellents, everything. So that was the bit brief discussion about all the 5 lectures. Now we are going ahead with the question and we will discuss one by one. Okay, so we are moving ahead with the questions. So Dash is a reflex arc that provides direct communication between sensory and motor neurons innervating the muscle. Dash is a reflex arc that provides direct communication. Okay. So what is the way that provide direct communication between sensory and motor neuron is the question. Option A, monosynaptic reflex. Option B, bisynaptic reflex. Option C, polysynaptic reflex. Option D, reflex. So as usual, we will wait for 1 minute. Now it's 6:15. At 6:16, we'll discuss the answer. I'm really sorry. Hi, Harini. Yeah. Can you please mute yourself and please type the answer in the chat box? Okay. Did you hear the question or you just now told? Then I can repeat the question. Can you mute yourself, please? I'm audible, right? Feel it. Yeah. So the correct answer is since it's a direct communication between sensory and motor neuron. So the correct answer is monosynaptic reflex. Okay. Sometimes it's also called as muscle stretch reflex, deep tendon reflex, which is a reflex arc that provide direct communication between sensory and motor neuron innervating the muscle. So that's about first question. Second question. The reflex arc involving the presence of one or more interneuron is called option A monosynaptic reflex, option B bisynaptic reflex, option C polysynaptic reflex, option D reflex. Now there is presence of one or more interneuron. So what kind of reflex it will be? That is the question. So 618 at 619 we'll discuss. Presence of one or more interneuron. So it's 619. So since the presence of one or more interneuron is called is there, so that synapse will be polysynaptic reflex. Option C is the correct answer. Okay. So the withdrawal reflex is polysynaptic, meaning that in addition to the sensor and motor neuron, this resources utilizes interneuron which pass signals between sensory and motor neurons, ultimately creating multiple synaptic connections. Next question. So this is the figure you can see, right? Monosynaptic reflex, direct communication between sensory and motor neuron. In polysynaptic reflex, we can see there is a purple color interneuron present in between sensory and motor neuron. Such one is called polysynaptic reflex. If it's direct connection just between sensory and motor neuron, then it is monosynaptic reflex or stretch reflex arc circuits. Next question. The stretch reflex is also called option A bistatic reflex, option B myotactic reflex, option C reflex, option D tendinitis. What is the correct answer? Stretch reflex is also called. Now we have just seen one another name. So this is one more question. Stretch reflex is also one another name of stretch reflexes. 620. So 1 minute at 621 we'll discuss. Okay. So the correct answer is myotactic reflex. Okay. Stretch reflex or myotactic reflex refers to contraction of muscle in response to. So myo means muscle. So that way so myotactic reflex means it's a contraction of muscle in response to its passive stretching. So the activity of muscle in response to the stretching is called as myotactic reflex. When a muscle is stretched, the stretch reflex regulates the length of the muscle automatically by increasing its contractibility as long as the stretch is within the physiological limits. Because as we know, there is some stretch sensors or sensors that can detect the change in length, which is called interfusal fibers. And all the further activity will happen as we discussed earlier. So remember, myotactic reflex is another name for stretch reflex. Physiologically, testing stretch reflex is an important test when we go to a doctor because option A, it is used to measure blood pressure. Option B, it helps to understand body temperature without using a thermometer. Option C, it correlates to pathology of central or peripheral nervous system. Option D, it helps to measure your walking speed. Physiologically, testing stretch reflex is an important test. So we have to physiologically test the stretch reflex. Why it is important to test stretch reflex? That is that will be the correct answer. What do you think is the correct answer for this question from the option? So now time is 6:24. At 6:25, we'll discuss the answer. Correct answer is C, right? It correlates to the pathology of central or peripheral nervous system. Why? Because the patellar reflex or knee jerk is an example of stretch reflex and it is used to determine the sensitivity of stretch reflex. Reflexes can be tested as part of a neurological examination, often if there is an injury to the central nervous system. To test the reflex, what doctors usually do, they do the knee jerk. So they will do the like one beating at the jerk and they check there is knee is getting jerked. So this is important to check whether the neurological activity is fine or not. To test the reflex, the muscle should be in neutral position. The muscle being tested needs to be reflex for the clinician to locate the tendon. After the muscle is relaxed, the clinician strikes the tendon. The response should be contraction of the muscle. If this is the knee jerk reflex, the clinician should observe a kick. So that's why knee jerk, the jerk should be there. The clinician rates the response. So what is knee jerk is one of the example of such kind of evaluation and that is used to check the pathology of central or peripheral nervous system. Question 5. Dash innervate extrafusal fiber and dash innervate intrafusal fibers. Option A, alpha motor neuron and alpha motor neuron. Option B, alpha motor neuron and gamma motor neuron. Option C, gamma motor neuron and gamma motor neuron. Option D, gamma motor neuron innervate extrafusal and alpha motor neuron innervate intrafusal. So what is the correct option? That is the question. 6:26 at 6:27 we'll discuss the answer. Okay. So correct answer is what innervate extrafusal? Alpha innervate extrafusal and gamma innervate intrafusal. So correct option will be option B. Okay. Motor neuron are divided into two groups, alpha and gamma, where alpha innervate extrafusal fibers and the highly contracting fibers that supply the muscle with its power. Gamma motor neuron innervate intrafusal fibers which contract only slightly. So contraction and the most abundant amount of fibers are extrafusal fibers. So that is connected with alpha and gamma detect. And sir, interestingly said when he taught this portion, we don't even know how much information, how who is telling the muscle, okay, this much amount of information go through alpha and this much amount go through gamma. So everything about the body is so fascinating, and science is growing in such a way that we are finding out what, like, everything is a, when we look deep into each and everything, we'll get that excitement and amazing of each topic, how beautifully our body, even each simple part is designed. So this is the figure of alpha innervating extrafusal and gamma innervating intrafusal. Next question. At the neuromuscular junction, the synapse is formed between presynaptic dash and postsynaptic dash. So you have to find what is presynaptic and what is postsynaptic. Okay. Option A, muscle presynaptic, motor neuron, postsynaptic. Option B, both muscles are presynaptic and postsynaptic. Option C, motor neuron, both motor neuron are pre and postsynaptic. Option D, motor neuron, presynaptic and muscles postsynaptic. What is the correct answer? So this is about a neuromuscular junction. So what will be the correct answer? So now it's 6:20 time. We'll discuss the answer at 6:30. Okay. So it's 6:30. Correct answer is presynaptic is neuron, right? Presynaptic is motor neuron and postsynaptic is muscle. So correct answer will be option D. So for convenience and understanding, neuromuscular junction or NMJ can be divided into three main parts. Presynaptic part, which is the neuron part or near terminal part. Postsynaptic part, which with receptors, the neurotransmitters release and get binding to that part, which is the motor end plate, and an area between nerve terminal and motor synaptic plate, that space. So that space between the presynaptic and postsynaptic neuron are called synaptic cleft. So correct answer is option D. Inactivation or blocking of acetylcholine receptors on muscle would cause. So someone, if we are trying to block the acetylcholine receptors on muscle, what will happen? Option A, overstimulation of muscle or tetanic response. Option B, no muscle stimulation and movement at all may lead to paralysis. Option C, normal muscle activity, no change is caused. Option D, both A and B. Inactivation or blocking of acetylcholine receptors on muscle would cause. So now acetylcholine is blocked and no acetylcholine is binding. So in that case, what will happen? So at 6:34, we'll discuss the answer. Okay. What is the correct answer? Correct answer is option B. Since acetylcholine is not coming, so the muscle cannot take any information from neuron to stimulate or any to do any movement. So that will leads to ceasing of the movement, which leads to finally may can leads to paralysis. Right? So usually there is a type of drug which neuromuscular blocking drugs, or it is also called as NMBAs, blocking agents. So they can block actually transmission at neuromuscular junction, which causes paralysis of the affected skeletal muscle. So this is actually accomplished via their action on postsynaptic acetylcholine receptor. So they go and bind. First, they go and bind with the post-acetylcholine all receptors. So when the real acetylcholine comes, already there is some component binded to the postsynaptic receptor. So there is nothing we can go. They can go and bind with the original receptors. As a result, muscle activity is ceased and it can lead to paralysis. Next question. An excessive increase in the release of acetylcholine receptors at the neuromuscular junction would cause an excessive increase in the release of acetylcholine receptor is the question. Option A, overstimulation of muscle or tetanic response. Option B, no muscle stimulation and movement at may all leads to paralysis. Option C, normal muscle activity, no change is called. Option D, both A and B. Now it is not blocked. Now the question is about what is the excessive activity of acetylcholine receptors will leads to. So the question is exactly opposite. Maybe answer can be also opposite. So 1 minute, 6:35 at 6:36 we will discuss the answer. So correct answer is A, right? Overstimulation of muscle or tetanic response. Because it's overly excited. So definitely more acetylcholine coming and coming and coming and muscle is keep on doing the its activity. So overstimulation of muscle or tetanic response will be the result. Which part of the ear contains the bones malleus, ingus and stapes? So this is in the figure malleus, ingus and stapes. So question is what is actually this part of the ear? Is it option A outer ear, option B middle ear, option C inner ear, option D there are no bones in the ear? What is the correct answer? Hello. So correct answer is middle ear. Middle ear contain the soft bones of ear, malleus, ingus, and stapes. So correct answer is option B, right? So it has another names. Malleus is also called as hammer. Incus is also called as anvil, and stapes also has the name of stirrup. Okay. Eardrums keeps the bones away from the outer ear. 10th question. Dash a structure made of calcium carbonate in ear that sends gravity, tilt, acceleration. So again, it's a question from ears. Dash are the structures made of calcium carbonate in ears. So which can sense gravity, tilt, and acceleration. Option A, hair cells. Option B, scale. Option C, bone. Option D, otolith. What is the correct answer? 6:38. So we'll discuss the answer in 6:39. So it is 6:39. Correct answer is otolith. Okay, otoliths are the structure that is made of calcium carbonate. Otolith has also other names such as taconium or otoconium. It's present in the saccule or utricle of the inner ear part. So basically, it is specifically present in the vestibular system of vertebrate. Saccule and utricle in turn together make an otolith organ. So if you are again going and seeing inside otolith, we can see two other things that is called as saccule and utricle. So what they do, they allows an organism, including humans, to perceive linear acceleration both horizontally and vertically. They have been identified in both extinct and extant vertebrates. So basically, they are the structures that maintain the balance. Okay. So that's why they can perceive linear acceleration both horizontally and vertically. Sometimes when there is some balancing issue and all, we can see like that must be the issue of inner ear, like some parts. So pressure variation or something. So doctors treat ear, we should go to the ENT doctor in that case. That because inside that part only the balancing part is also present. Some more questions. Okay, next question. Which of the following cell type is part of the peripheral nervous system? Option A, cortical neuron. Option B, oligodendrocyte. Option C, microglia. Option D, Schwann cell. So it's a question regarding peripheral nervous system. Some part of the cells are in central nervous system and others are in peripheral nervous system. So in the options given below, one we can see in peripheral nervous system. What are that types of cell is the correct answer. Will be the correct answer. So at 6:42, we'll discuss the answer. So it's 6:49. What is the cells that are present in glial cells that is covering the peripheral nervous system axonal part? They are Schwann cells, right? Oligodendrocyte is in central nervous system and Schwann cells are present in peripheral nervous system. In almost all the weeks, we are seeing this question multiple ways in multiple ways. So please Remember that oligodendrocytes are in CNS and Schwann cells are in PNS. Okay. So they play a role in survival and function of neuron. In response to nerve injury, Schwann cells undergo rapid changes in phenotypes and their basal lamina provides a conduit for axon regrowth, a critical process for nerve regeneration. So Schwann cells are important in some kinds of injury since their lamina can provide a conduit for axonal regrowth, which is required in neuronal regeneration. Next question. Which of the following best describes the order of signal conduction in the stretch reflex arc? Option A, sensory neuron, spinal cord, motor neuron, effector. Option B, sensory neuron, spinal cord, frontal lobe, motor neuron, effector. Option C, motor neuron, spinal cord, frontal lobe, sensory neuron, effector. Option D, sensory neuron, effector. So what is the correct answer is the question. Correct pathway of signal conduction in stretch reflex arc. Where it starts, where it goes through, where it reaches, where it ends. If you know that answer, that will be the correct answer. So please read the option. It's 6:43. I will wait till 6:44. Then we'll discuss the answer. So what is the correct answer? So it is 6:44. Yeah, so it starts with sensory neuron, right? Sensory neuron takes the information from the sensor, give it to the, so it must be a polysynaptic reflex. That's why interneuron is there. So it will give to the spinal cord or interneuron. Then that will give information to the motor neuron through the ventral horn. They will reach the effector. So correct option is option A, right? No need of discussion, I guess. So this is the pathway. Muscle spindle senses the stretch. So remember, muscle spindle or interfusal fibers dictate the change in length. Sensory neuron conducts action potential. 3. Sensory neuron synapses with alpha motor neuron and inner interneuron. Alpha motor neuron to quadriceps is stimulated while the alpha motor neuron to the hamstring is inhibited. Finally, what will happen? Quadriceps contracts and hamstrings relax. So this is one of the stretch reflex arc example. So please remember, starting with sensor, which is interfusal fibers, giving to sensory neuron, giving it to the interneuron or spinal cord, reaching there and passing it to the alpha motor neuron. So then alpha motor neuron, finally it will leads the information to the effector. Next question. Muscle spindle is a dash and that detects change in dash. Just now we discussed, right? Muscle spindle is what and what detects change in dash. This same question we discussed in the live session of skeletal system also, that time also. So options are muscle spindle is a options A neuron that detect change in the concentration of ions in extracellular fluid. Option B again it's a neuron and detect change in the concentration of ions in intracellular fluid. Option C it's a stretch receptor and detect change in length of muscle. Option D it's a stretch reflex circuit and it can detect change in conduction velocity of muscle neuron. So what is the correct answer? A, B, C or D? A, B, C or D? So muscle spindle is a dash and that can detect in dash. So it's 6:46. At 6:47 we'll discuss. Easy, I guess. So what is the correct answer? Correct answer is option C. It's a stretch receptor and it can detect change in the length of the muscle, right? It's a intrafusal fibers, muscle spindle, another name is intrafusal fiber. And they are the sensors that can detect change in the length of the muscle. So it's a part of muscle only, not a neuron. There is some neuron coming and connecting to it. That doesn't mean it's a part of neuron. It's a part of muscle only. So it's a stretch receptor and it can detect the change in length of the muscle. Option C is the correct answer. Yeah, again, please remember this figure. Okay, this is the muscle spindle. So it is getting passing and again coming and which of the following muscle type is present in a large number? So it is about comparison, which is higher in number is extrafusal or intrafusal or both are equal in number is the question. So which is higher in number? Option A, extrafusal fiber. Option B, intrafusal fiber. Option C, both are present in equal number. Option D, none of the above. So what is, which muscle type is present in large number? Is the question. 648, okay. Yeah. So what is present in higher number? We know intrafusal muscle fibers, we just detected it are muscle sensors. But if what muscle type have the highest power of contraction, if they want to do the contraction activity, that class of muscle fibers are extrafusal fibers. So definitely extrafusal fibers should be large in number than intrafusal fibers. Because intrafusal are just sensors, so they are small in number and they can detect the change in length. But in order to respond and do the highly contraction activities, extrafusal should be highly present. So highly present or largely present will be extrafusal fiber. Okay. Even in all the images itself, we can understand. Compared to intrafusal, extrafusals are very high. Next question. Which of the following is or are sensors? Option A, extrafusal fiber. Option B, intrafusal fiber. Option C, both A and B. Option D, none of the above. I think we discussed in here from SAR from week 8, lecture 2. Lecture 2 in week 8, SAR was discussing this. What are or which is or are sensors in the option? So which is R or are sensors? Just now also we discussed. So what will be the correct answer? 651. Harini, please type the answer. If it's right or wrong, doesn't matter. We anyway discuss. So correct answer is intrafusal fiber, right? Intrafusal fiber is a sensor which can detect change in the length. So correct answer is option B. Now again, it's about extrafusal, intrafusal, what type of neuron are connecting to each. Okay, that is very important. Please remember, find a clue to connect it to what connects what, then it will be easy for you guys. Alpha motor neuron synapses on dash and gamma motor neuron synapses on dash. Option A, extrafusal fiber, extrafusal fiber. Option B, extrafusal fiber, intrafusal fiber. Option C, intrafusal fiber, extrafusal fiber. Option D, intrafusal fiber, intrafusal fiber. So alpha motor neuron connect what and gamma motor neuron connects to what? That is the question. I think this is also second question we are asking regarding the same type of question. Maybe questions can be asked opposited. Extrafusal fibers are innervated by what kind of neuron? Intrafusal fibers are innervated by what kind of neuron? Otherwise, alpha motor neuron. Synapses on what kind of muscle? So questions can be asked in any way. So what is the correct answer? Alpha motor neuron synapses on dash and gamma motor neuron synapses on dash. So it's 6:52 or 6:53 we'll discuss. Alpha gamma. So alpha is the first letter. So it must be connecting to the one fiber that is present in large number. Next clue. So alpha is connecting to extrafusal fibers and gamma is connecting to smaller in number, but they are the sensors which are intrafusal fibers. So from the option, correct option will be option B. Okay. Alpha is connecting with extra and gamma is connecting to intra. Also, like one of the last week after discussion of week 5, when the assignment solution released, I told that week 5 there is some wrong answers. So today they have like retyped that the corrected option and they have corrected the assignment mark also. So please go and check. Okay. Which of the following is a part of inner ear? Option A, ear canal. Option B, stapes. Option C, eardrum. Option D, cochlea. Which of the part is a inner ear? Earlier there was a question regarding middle ear. Now the question is regarding inner ear. What is a part of the inner ear? So 6:54 at 6:55 we'll discuss again. Correct answer is cochlea, right? Cochlea is a part of inner ear. What are the other options? Let's go by one. Ear canal is a part of external ear. Stapes is part of middle ear. Ear drum. Ear drum is also part of what ear? Ear drum. Tympanum. Okay. So tympanum is also a part of middle ear. External ear and ear canal is part of external ear. Malleus ingus stapes bones are part of middle ear. And cochlea, otolith, all these things are part of inner ear. Okay. So that's about cochlea. Cochlea is a fluid-filled spiral spaced cavity found in the inner ear that plays a vital role in sense of hearing and participate in process of auditory transduction. Sound waves are transduced into electrical impulse that the brain can interpret as individual sound frequencies. So that is the correct answer. Next question. Of the vestibular hair cells in ear, which of the following is taller? Again, that taller part question comes. Please remember when Sir draw the image, he labeled it wrongly. But whatever he labeled it is wrongly, but we discuss the answer now. But taller one is what? Among the hair cells, taller one is very less in number, only one is there, but there are lots of smaller hair cells present. So what is taller one and what is smaller one? Taller one, option A, gynoecilia, option B, stereocilia, option C, deprocilia, option D, heliocilia. What is the correct answer? I gave a clue also, whatever the labeling was wrong. So if you remember that thing, then you can say. So correct answer is gynoecilia is the taller one. Okay, gynoecilia. Stereocilias are the smaller ones, but when are labeled stereocilia as the longer, taller one. But no, gynoecilia is the taller one, and stereocilias are small and abundant in number. Yeah, this is the figure. So this blue one or this pink one present in the purple one. That is the longer one is gynocilium and all the smaller ones are stereocilium. Of the vestibular hair cells in inner ear, which of the following are more in number? Option A gynocilia, option B stereocilia, option C deprocilia, option D heliocilia. More in number is the question. Okay, more in number. Now, just now we saw the answer. So it's easy, I guess. More in number. If taller one is gynoecilia, then more in number will be the other one. Smaller and more in number. So correct answer is, now it's 6:59. There is 1 minute. At 7 we'll discuss. Correct answer is stereocilias are more in number. Okay, the same figure you can refer to. Next question, otolith present in ear is made up of. This we have seen just now one another question in an another. What is the structure that is made of something? So what is that something? Option A hydroxyapatite, option B potassium oxalate, option C sodium chloride, option D calcium carbonate. What is the correct answer? Otolith teeth is present in ear is made up of dash. Correct answer is calcium carbonate. Right? Statoconium or autoconium is are the other names. It's made up of calcium carbonate structure, and inside that we can see two saccule or utricle in the inner ear. So these are dealing with the vestibular system of the vertebrate, which is for the valency. Major population of skeletal muscle is dash. Again, same question. More in number, major population, everything is same only. Option A1 intrafusal fiber, option 2 extrafusal fiber, option 3 both A and B are equal, option 4 none of the above. It's 7-1 at 7-2. This we have discussed now multiple times. I know you guys know the answer. Major population of skeletal muscle is extrafusal fiber. Correct answer is option 2. They are responsible for generating force and movement and the predominant type of muscle in the skeletal muscle. At neuromuscular junction, muscle is dash and neuron is dash. Just before this, we have seen what is presynaptic and what is postsynaptic. Now they are asking muscle is what and neuron is what. Questions just opposite. Option 1 postsynaptic, postsynaptic. Option 2 both are presynaptic. Option 3 muscle is presynaptic and neuron is postsynaptic. Option 4 muscle is postsynaptic and neuron is presynaptic. What will be the correct answer? So correct answer is neuron is presynaptic and muscle is postsynaptic. So correct answer is option 3. Sorry, muscle is postsynaptic and neuron is presynaptic. We are getting information from neuron and giving it to the muscle. So neuron is presynaptic and muscle is postsynaptic. Right? Postsynaptic means part of the muscle cell that receive the signal from the neuron. So from the neuron, it's receiving muscle is receiving the signal. Presynaptic means where the information is, who is giving the information. So neuron is giving the information and sending the signal across the axon terminal to the skeletal muscle. So presynaptic is neuron and postsynaptic is the muscle. Fourth is the correct option. RYR and DHPR are dash in muscle. Option 1 potassium channel, option 2 calcium sensors, option 3 chlorine ions, option 4 none of the above. RYR and DHPR are dash in the muscle. So now pyridine dihydropyridine receptor. Sir told the full form also. So what will be the correct answer? So correct answer is they are calcium sensors which is connected with sarcoplasmic reticulum. So whenever the motor neuron innervate and acetylcholine release, they can get the acetylcholine which will again tell the sarcoplasmic reticulum to open the calcium channels and thus they are calcium sensors. So ryanodine receptor is a calcium release channel located in the sarcoplasmic reticulum of muscle cell, responsible for releasing calcium into cytoplasm when triggered by a signal from dihydropyridine receptor. So first, who is getting the information? DHPR. DHPR tells RYR, which is situated in the sarcoplasmic reticulum, to make open the calcium release. So DHPR is a voltage gated calcium channel located on the muscle cell membrane which acts as a voltage sensor detecting changes in membrane potential and triggering the opening of the RYR to release calcium from SR. Okay. In muscle, calcium is released from dash. Just now we discussed. Option 1 neuron, option 2 sarcoplasmic reticulum, option 3 cytoplasmic reticulum, option 4 all the above. What is the correct answer? Calcium is released from. Muscle is also written. So from muscle, calcium is released from. In muscle, calcium is released from sarcoplasmic reticulum. Option 2 is the correct answer. Okay. So it's a specialized organelle within muscle. It's a cytoplasmic reticulum. Since it's in muscle, it is known as sarcoplasmic reticulum. Anything connecting with muscle, sarcoplasm, cytoplasm is known as sarcoplasm. So everything connecting with muscle is known as sarco. That word is for connecting with that part. Okay. So this is a organelle present in muscle which store calcium ion. When a muscle needs to contract, calcium is released from the sarcoplasmic reticulum, triggering muscle contraction. We have seen sliding filament theory. So this is sarcolemma. Membrane is known as sarcolemma. Sarcoplasmic reticulum is the endoplasmic reticulum of muscle. Terminal cisternae, T-tubule and triad. Which of these is not a bone in ear? Option 1 malleus, option 2 stapes, option 3 femur, option 4 ingus. All these are bones, but one is not a part of ear bone. So what is not a part of ear bone among the option is the question. So it is 7, 8. Now at 7, 9 we'll discuss. So that is not a part of bone in ear is femur. Femur is a part of our thigh bone. Okay, it is the longest bone present in our body. The other three, malleus, ingus, stapes, are part of the middle ear, collectively called as ossicles. Okay, they are responsible for transmitting the sound vibration which is passed through the ear canal to the inner ear or cochlea. So femur, in the other way, femur is the longest bone which is located in thigh. It's not a part of ear structure. Again, about hair cells. Long hair is called that one long hair in the hair cell. Option 1 stapes, option 2 femur, option 3 gynoecilia, option 4 stereocilia. Which is the longer hair cell? This also we discussed multiple times. What is the correct answer? One long hair is called Ctenocelia is the correct answer. Ctenocelia. Okay. So one long non-sensory hair called ctenocoelium and many smaller actin-based hairs called stereocilia, which are the primary sensors for sound and balance. While stereocilia are the main functional hairs that bend to create signal, ctenocoelium is crucial for the proper arrangement and orientation of the stereocilia. So stereocilia are the main players. They are the vibrating in what direction the wave is receiving, and according to the movement, they are playing a major role. But kinocilium is required to maintaining the proper arrangement and orientation. Again, the same question. Hair cells in ear have multiple short hair called multiple short hair. What is that multiple short hair? Correct answer is stereocilia, right? Multiple short hair is, we discussed now multiple times. So I'm not explaining. Stereocilia are multiple and short and in large in number present among hair cells. Which of these is cell is related to olfaction? Now it's a question related to olfaction. Option 1 hair cell, option 2 hippocampal cells, option 3 motor neuron, option 4 mitral cells. What is related to olfaction is the question. Olfaction. It is 7:11. At 7:12, we'll discuss the answer. Correct answer is mitral cells. Okay, mitral cells. I'll show one image so you'll understand. So they are basically present in olfactory bulb. Olfactory bulb is the primary processing center for olfactory information. So first they will receive information from olfactory receptors, and then they will transmit the odor information to high brain region. So see, these are the receptors. So they are connected to the receptor. From mitral cells will receive information from olfactory receptors present in olfactory epithelium. From there they will give the information to the neuron, right? Axons of mitral cell. They will give the information to the brain center of olfaction. Now it's a problem, interesting problem. 1 decibel represents an actual increase of dash times in sound energy. 1 decibel represents an actual increase of dash times in sound energy. Option 1 0.126, option 2 1.26, option 3 12.6, option 4 126. What will be the correct answer? It's based on an equation. So I will show the equation and discuss the answer. So at 7:14 we'll start discussing the answer. Okay. Till then you can guess what is the correct, what will be the correct answer, why it is a correct answer. Yeah. So correct answer is 1.26 times. Why? There is a equation dB is equal to 10 log 10 S1 by S2, where S1 and S2 are the intensity of two sound waves. So here they are saying 1 decibel. So dB instead, let me write for you guys. Yeah. dB is equal to 1, right? Now we will put 1 is equal to in the equation 10 log 10 S1 by S2, right? Ah. Now what is the answer? So log 10 S1 by or P1 by P2. If it's pressure wave, then we also put it as P1 by P2. So log 10 S1 by S2 is equal to 1 by 10, right? Which is 0.1. Now we are taking anti-log on both sides. So it will be is equal to, so if we are taking anti-log, then it will be S1 by S2, right? So S1 by S2 is equal to, anti-log will be base to the 10, right? So 10 raised to 0.1. 1 by 10, anti-log will be raised to 0.1. That will be approximately is equal to 1.258 6, which is equal to 1.26 from the option. So this is the correct answer. How did it come? This is based on this equation. Okay. So dB is equal to 10 log 10 S1 by S2 is the equation. So please remember it. Next question. Cochlea is a part of dash. Option 1 inner ear, option 2 middle ear, option 3 outer ear, option 4 none of the above. So what is the correct answer? Cochlea is a part of. This also we discussed multiple times now. So it's 7:17. At 7:18 we'll discuss. Cochlea is a part of dash. So cochlea is a part of inner ear, right? We know cochlea is a part of inner ear. The spiral shaped cavity is responsible for converting sound waves into neuroimpulses that are sent to the brain, allowing us to hear. So now we have seen multiple questions from the week 8. Let's see if you guys remember week 7, which was we learned about multiple disorders, neurodegenerative disorders like Alzheimer's disease, which there is a memory disorder, Parkinson disease, which is a motor disorder, then amyotrophic lateral sclerosis, about spinal cord injury. All this we have seen. So let's see some more questions regarding which you remember or not. Then we'll wind up today's session. Which part of the brain is majorly affected by Alzheimer's disease? Option A, substantia nigra. Option B, hypothalamus. Option C, hippocampus. Option D, pre-Botzinger complex. This we have discussed multiple times. I told you guys, please remember what disorder affecting which part, which kind of neuron is present there, what kind of disorder is that, what is the characteristics of disorder. So correct answer is hippocampus, right? Hippocampus is the structure, is the memory center of the brain. So it's Alzheimer's is a memory disorder and it starts from the hippocampal region. So it's a disorder that slowly destroys memory and thinking skills and eventually ability to carry out simple tasks. Initially takes place in brain involved in memory, including the entorhinal cortex and hippocampus. Later it will lead to cerebral cortex. That is the stage when they will start, the patients will start losing the memory of language, reasoning, social behavior, everything. So first starts with hippocampus, then entorhinal cortex, finally to cerebral cortex. What happens to neurons in Alzheimer's disease? Option A, cells bodies become larger. Option B, cells become starts to die off first. Option C, processes become demyelinated but alive while cell bodies die off. Option D, processes starts to die off. What is dying first when? Correct answer is processes starts to die off, right? Option D is the correct answer. Due to the building up of the proteins like tau, amyloid beta, that's as a result, processes will start dying off first. CA1, CA2, DG are part of which part of the brain? Option A, substantia nigra. Option B, hypothalamus. Option C, hippocampus. Option D, pre-Botzinger complex. What is the correct answer? CA1, CA2 and DG. So it's 7:21. At 7:22 we'll discuss the answer. 30 seconds more. CA1, CA2 and DG are part of hippocampus. Remember the structure like this. Below that there is dendrite gyrus where we can see stem cell progenitors of neurons present. So they have a head, body and tail. Head is expanded part, and tail is the thin curved part. Based upon histology, hippocampus proper or cono ammonis is divided into C. That is, cono ammonis is the CA1. CA stands for CA2, CA3, and CA4. Which of the following is not a neurotransmitter? Option A, islets of Langerhans. Option B, serotonin. Option C, GABA. Option D, glutamic acid. Not a neurotransmitter is the question. That means three are neurotransmitters. There are different types of neurotransmitter: excitatory, inhibitory, and one among the option is not a neurotransmitter. This we have discussed, I guess, in the last week. Correct answer is islets of Langerhans are not a neurotransmitter. They are part of pancreas, but all others, serotonin, GABA, and glutamic acids, are neurotransmitters. Right? So islets of Langerhans is not a part of not a neurotransmitter. It's a part present in the pancreas. Neurons of which of the following area is majorly affected by Parkinson disease? Now the question is regarding Parkinson for motor disorder. So which part is getting affected? Option A, substantia nigra. Option B, hypothalamus. Option C, hippocampus. Option D, pre-Boxender complex. What is the correct answer? Correct answer is substantia nigra. Right? It's a midbrain dopaminergic nucleus which has critical role in modulating motor movement and reward function as part of basal ganglia circuitry. Projection from substantia nigra to putamen is called nigrostriatal pathway, which are critically involved in the motor deficit observed in Parkinson disease. So just now I said Parkinson disease is a dash disorder. So what activity is getting affected? Option A, memory. Option B, motor activity. Option C, hearing. Option D, vision. So correct answer is motor activity. Okay, it's about a movement disorder. So different patient have in different terms. It's depending on how severe the disorder is, what stage it is. But it's mainly get affected. Motor activity is getting affected. Which function is lost in Alzheimer's disease? If Parkinson is a motor activity disorder, then what is getting lost in Alzheimer's disease? Option A, memory. Option B, motor activity. Option C, hearing. Option D, vision. Which function is getting lost in Alzheimer's disease? Correct answer is memory. We discussed, right? I don't think we have to discuss again. Yeah, so that is the last question. So please go and see the assignment 5 and assignment 2 solution because there were two wrong answers. They have released whatever the answer that was wrong in assignment number 5, I think question number 2 and 9 or something. And in question assignment number 2, the 10th solution was wrong. So please, today they have revised the answer and revised the assignment questions. So please go and see it and please try to attempt all assignment questions. Now week 9 sessions class is also released, so please hear it and please try to join for the live session also. I know the videos are available in YouTube, but still it will be better in an interactive session. So yeah, that's it. See you in next class, which is week 9 discussion. We'll be going ahead with the other sensory systems in brain. Then after finishing with the brain neural system and all, we are moving ahead with the next type of um, organ system. Okay, now we have seen integumentary system, skeletal system. Still nervous system is going on. It will go for next week some part also. Then we'll be going ahead to the other types of organ system. So that is it. So see you. Bye bye. Take care.